Hi, everybody. It's your AP Biology teacher here, Mr. Poser. Today we're getting into topic 3.5, which is on cellular respiration. This is the last topic of Unit 3, and it's a big one. All right, this is one that comes up on the AP exam for sure in some capacity. It's usually in the FRQ section. It's a big deal, so I don't want to waste any time, and let's get into it right away. What is cellular respiration? I've been referring back to this quite a bit in the previous topic videos. It is the process by which macromolecules, namely carbohydrates like glucose, for example, are used to synthesize ATP using coordinated enzyme-mediated reactions, and they are found, and this is found in all forms of life. All living things need to have ATP to drive their cellular processes, and all living things do this to an extent. All right, there's three main phases that I'm going to be referring to, and we're going to get into more details about later in the video. When it comes to cellular respiration, there's glycolysis right over here. There's the Krebs cycle, and there's what's called the electron transport chain. You can see one of them occurs outside of the mitochondrion, and two of them occur within the mitochondrion. And we're going to get into detail about how these work in just a second. But first, we have to get into the anatomy of the mitochondrion, the powerhouse of the cell. Why? Because different processes happen in different parts of the mitochondrion. It's compartmentalized. Remember, we talked about that in the last unit, how the origin of the mitochondrion has to do with being a free-living aerobic prokaryote that got swallowed up by another cell, and they live together. So anyway, the mitochondrion is its own kind of free-living organism in a way, all right? But we got to know how the anatomy of it to figure out how cellular respiration works a little bit. All right, we don't have to know too much. It's only 4. All right, so if you can remember, the mitochondrion has 2 membranes, right? It has an outer membrane that's smooth, right, from the ancient prokaryote swallowing it up, right? The vesicle, basically. It has a smooth outer membrane. It has an intermembrane space, all right? Inter means between, because, well, it has 2 membranes, and the inner membrane is all folded up like this. It's highly convoluted, all right? And this is the location of phase 3, the electron transport chain, that we're going to look at in detail here in a little bit. So it has a highly folded inner membrane and a smooth outer membrane, all right? And why is that inner membrane all folded up? Well, lots of folds increases surface area. Lots of surface area gives us lots of space for lots of reactions, and lots of space for lots of reactions gives us more ATP. So as I put down here, more inner membrane folds equals more surface area and more ATP. All right, and then finally, the inside of that inner membrane is what we call the matrix. And that is the location of the Krebs cycle, and that will become relevant here in the electron transport chain as well once we get into the specifics about that. All right, so the Krebs cycle is phase 2 of the cellular respiration process, and that's what we're going to look at here. Now, speaking of the Krebs cycle, glycolysis and the Krebs cycle, all right, the whole point of cellular respiration is to make ATP, right? But those first 2 phases, they're primarily responsible for producing 2 molecules, NADH and FADH2, okay, which are known as reducing agents. Now, in the last video, we looked at NADPH, all right, which I told you is basically just an electron carrier, and we're going to get into the specifics of how that works here in just a second, all right? They're basically just carrying an electron from one place to another to start an electron transport chain, similar to what we saw during the light reactions in our last topic, all right? So NADH and FADH2 are reducing agents. They reduce other molecules by transferring an electron to them, right? So they're going to carry an electron from one place to another, transfer it from one molecule to another, and that's basically it, all right? And an agent is not like, I'm a secret agent, right? It's just something that does an action, okay? And in this case, it reduces another molecule, and I'm going to show you what that means here with a little interactive that I made. All right, so reduction, all right, is a process of gaining an electron. Oxidation is a process of losing an electron. So I'm going to show you a super simple example of a redox reaction, as we call it, or an oxidation reduction reaction involving NADH here, which is one of our 3, reducing agents that we're going to be talking about. All right, so when NADH doesn't have an electron, it has a little positive charge, and we call it NAD+, all right? So remember, electrons are negatively charged, so if this is lacking an electron, it's slightly positively charged, right? So it's NAD+. But once it gains that electron, Okay, it is what's called reduced. Okay, so reducing something means lowering its overall charge. All right, so when it gains an electron, the overall charge of that molecule is reduced. Okay, so think about you're making it more negative. If you're adding something negative, you're subtracting, right? So that's what's called reduction. So when NAD plus is reduced, it gains an electron, it becomes NADH. All right, and now it's carrying an electron that it can transfer from one place to another. Okay, so here's a protein, generic protein over here. And what NADH can do is move on over here and transfer an electron. All right, and therefore, this NADH loses an electron, and this protein gains an electron. So now this protein is reduced. All right, it gains that electron. All right, and since this NADH over here lost its electron, it is what's called oxidized. Okay, so oxidation reduction reactions basically mean just transferring an electron from one thing to another. And that's pretty much it. Whatever's gaining the electron is reduced. Whatever's losing the electron is oxidized. And a way that you can remember that is oil rig over here. Oxidation is loss, OIL. Reduction is gain, RIG. Oil rig. Okay, so whatever's gaining an electron is reduced. Whatever's losing the electron is oxidized. Okay, and when NADPH transfers an electron, it's getting oxidized, and whatever it's transferring the electron to is getting reduced. Same thing with NADH and FADH2, and that's what glycolysis and Krebs cycle are going to be mostly producing here: NADH and FADH2, those electron transfer molecules. Okay, so now let's get into the specifics of those three different phases of cellular respiration. And remember, the first two, glycolysis and the Krebs cycle, were primarily focused on making NADH and FADH2. We're going to make some ATP as well, but, you know, you'll see. All right. In order for this whole thing to begin, we need glucose. All right. So glucose is a very, very central, important molecule here. Lots of different biochemical reactions, a lot of different metabolism pathways are so that, you know, you can make glucose. All right. And glucose can be used to make ATP. All right, so glycolysis is a series of reactions that's going to turn glucose into another molecule called pyruvate. All right, and by extension, what we're also is going to happen here through a series of enzyme-mediated reactions is produce some ATP, which is, you know, the main goal, right? We want to make ATP. That's the point of cellular respiration, and it's going to make some NADH, which is what, you know, the good stuff is here, and I'll show you why here in just a little bit. Okay, but it's turning glucose into pyruvate, and it has some byproducts, ATP and NADPH. Okay, so here's your formal definition. It is a metabolic pathway that produces 2 ATP and 2 NADH by converting glucose into pyruvate. I want to say it's like 6 or 7 reactions. I had them memorized at one point in my life, but I don't have them anymore. All right. So the important thing here is that it turns glucose into pyruvate, and it makes 2 ATP and an NADPH, and it makes this happen in the cytosol. So this actually doesn't happen in the mitochondrion. All right, so in prokaryotes, or anaerobic prokaryotes, I should say, prokaryotes that don't use the Krebs cycle and the electron transport chain, they're going to be basically relying on this, solely this, for ATP, just turning glucose into pyruvate and getting those 2 ATPs out of it. Yes, that's the goal, right, is to make ATP. So cells that don't have mitochondrion, if they're anaerobic, which means they can't do the rest of cellular respiration with oxygen, they're just going to be relying upon this. Okay, this is what's called fermentation. All right, if they're going to be making ATP without oxygen, okay, without doing this Krebs cycle and the electron transport chain that we're going to look at here in just a second, they are doing what's called fermentation. All right, in certain types of bacteria and things like yeast. Yeast is a single-celled organism. It's a type of fungus, actually, that relies upon using fermentation, just basically glycolysis, to make its ATP. All right, and that pyruvate either becomes ethanol, which is, you know, how brewing happens, or something like lactate or lactic acid. Okay, so when you're going into oxygen debt, actually, when your muscles are not getting enough oxygen and they still need ATP, they're going to rely on a similar process to this, lactic acid fermentation, to make ATP and to keep your muscles going, even though they're not getting enough oxygen. All right, I tell my cross-country athletes about this all the time. But yeah, different foods can be made through fermentation, like yogurt and, you know, beverages, and kimchi over here are all results of fermentation. Okay. So anyway, glycolysis here is phase one, and this is where no oxygen is involved just yet. All right. So we made some ATP and we made some NADH. All right. Now, if oxygen is present and you have the mitochondria, you have the powerhouse of the cell, what happens next is what's called the Krebs cycle. This is phase two. Pyruvate is shuffled into the Krebs cycle, and this is a cycle of reactions that, again, we don't have to memorize by any means here. And it makes more ATP. Yes, that's the goal, right? We want more ATP. But more importantly, I'm going to say it makes 6 NADH molecules and 2 FADH2, those reducing agents, those electron carriers, right? It's able to produce 8 of those molecules in this process over here, this series of cycle of reactions. All right, and as a byproduct, it produces carbon dioxide, which is consequently what you breathe out all the time. It is it's a byproduct, it's a waste product of cellular respiration. Why do you need to breathe out carbon dioxide? Well, because you don't need it. It's a waste product of this type of reaction. Okay, so the Krebs cycle, here's our formal definition. It's a cycle of reactions where pyruvate is used to reduce NAD plus to NADH and FAD to FADH2. So they're going to be both carrying electrons while releasing carbon dioxide as a byproduct. All right, and here's some specifics. There are enzyme-mediated reactions here, as you can see, and it's produces some ATP, it produces some carbon dioxide, and it produces some NADH and FADH2. And this occurs, cyclically here, so long as there's pyruvate being provided by glycolysis, right? And this is occurring in the mitochondrial matrix. So we are inside of the inner membrane. And the main thing that we're producing here, like I said, is the electron carriers, NADH and FADH2. And why is that important? Well, because we are starting the world famous, the ever so important mitochondrial electron transport chain. All right. And we made some ATP. All right. We made some ATP with these first two processes here, right? So glycolysis, we got 2 ATP. Cool. Right on. Yes. Krebs cycle, we can get another 2 ATP from that. All right. Nice. But with the electron transport chain, what the mitochondrion can do per glucose molecule is make 30 plus ATP molecules, which is highly, highly efficient, right? We can phosphorylate 30 ADPs into 30 ATPs just using the electrons in the electron transport chain that is brought over here from the NADH and the FADH2. All right. And consequently, as well, we are producing some water as a byproduct. And we're going to see where that comes from as well here in just a little bit. So here's your formal definition. We're going to get into a little bit of the details about the electron transport chain. It is a series of oxidations or reductions, redox reactions, passing of electrons, that establish a proton gradient across a membrane. All right. Very similar to what we saw in the last topic video with the light reactions. Couldn't think of what they were called for a second. Okay. But this is like making a proton gradient. It's like on steroids. All right. It's like there's so much going on here in terms of this proton gradient. All right. So we are establishing a proton gradient across the inner mitochondrial membrane in eukaryotes, okay, but it occurs along the plasma membrane in aerobic prokaryotes, in some prokaryotes that are able to do this, all right? So the prokaryotes that we talked about before, anaerobic prokaryotes means they don't use oxygen to make their ATP. They're just relying on glycolysis. But aerobic prokaryotes are going to be doing this process here that I'm going to show you along their plasma membrane. Okay, but in other eukaryotes, all right, we have mitochondria, and this occurs along the inner membrane. All right, the electron transport chain, the ETC, establishes a proton gradient that ATP synthase, yes, ATP synthase, uses to phosphorylate ADP into ATP in large amounts. And this whole process is what's called oxidative phosphorylation in the presence of oxygen, which we're going to see what it does here in just a second and why that's important. In the presence of oxygen, ATP synthase is going to use that proton gradient to phosphorylate ADP and make ADP into ATP. And by extension here, what this can also be used for is to generate heat and endothermic organisms. All right, so we are endothermic. We use our metabolism to maintain our body temperature. And this similar process here can be used to generate heat instead of making ATP as well, so that we can, maintain our body temperature so that our enzymes work properly, right? All right, but anyway, let's get into the specifics here of how this works. So where are we? We are in the mitochondrion right now. We are in Well, here's the intermembrane space. Here is the mitochondrial inner membrane, and here's the matrix. All right, so we're inside the mitochondria, and we have a system of proteins over here. Now, do we have to memorize what all these proteins are? No, we don't, okay? Well, we should get the gist of it here, okay? Here's my electron carrier. It's NADH over here. What is it going to do? Well, it's going to transfer an electron to this protein over here. And as it showed you before, it gets oxidized because it drops off its electron. It's doing its job, okay? And here's FADH2. Here's our other electron carrier. It's going to drop off an electron at protein number 2 over here and become oxidized as well. All right, so here's our two main our reducing agents over here, our main two products of the Krebs cycle. They're going to be dropping off some electrons over here. And just like what we saw in the last topic, all right, these electrons are going to be transferred. They're going to bounce and get passed along from protein to protein. And that energy is going to be used for, well, pumping protons. All right, so all three of these proteins over here, 1, 3, and 4, their job is to pump protons from the mitochondrial matrix into the intermembrane space, and they're going to use that, or they're going to do that using the energy from these electrons. All right, so these two reducing agents over here are bringing these electrons, and once they get passed over, ping, pong, 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 they're able to pump some protons out. All right, and therefore we are establishing a proton gradient. We're establishing a concentration gradient. These are being forced out of here, and diffusion says they want to come back in. And guess what? There is a way for them to do that. They have to go through ATP synthase in order for that to happen. Okay, so they are going to do chemiosmosis once again and pass from where there's high concentration to where there's low concentration. They want to come back in, but the way to do that is through ATP synthase. And just like before, ATP synthase is going to take some ADP, ADP meaning diphosphate, and an inorganic phosphate, and use that proton gradient to make some ATP. All right, and remember, this happens in large amounts. All right, this is continually cycling, and it can make 30 ATPs using one glucose molecule all the way from the beginning of glycolysis. All right. So now here's the thing though. All right, we're making our ATP, cool, but there's a little bit of a problem here that still needs to be solved. Now these electrons are like stuck here at protein 4 here, unless of course we have some oxygen. So what is oxygen all about? I've been talking about oxygen this entire time. None of this can happen without oxygen, and here's why. Ready? Oxygen is what we call the terminal electron acceptor. Okay. These electrons need to go somewhere, all right? They can't be sticking around here forever. They're going to block up this whole chain here. We need to pass along more electrons and power this whole process to make ATP, all right? So these electrons need to go somewhere. And what's going to happen is they're going to be picked up by oxygen. So the reason, guys, the reason you need to breathe oxygen is so that your oxygen molecules, oxygen gas molecules, can pick up electrons from your mitochondrion. All right, and make it safe for this to continue on here. All right, the oxygen is going to pick up those electrons and pick up some hydrogen ions that are also on the inside of the mitochondrial matrix. I mean, look at them all over here. All right, and it's going to form some water. So water is actually a byproduct of cellular respiration. It takes in those electrons from the electron transport chain with the help of oxygen. All right, that's why you need to breathe, you guys. And as soon as you stop breathing, this chain gets all bunked up. Okay, so here we are at the recap, all right? In case you missed anything, I put it all in writing, or in case I missed anything, here it is. Cellular respiration uses macromolecules, carbohydrates, to synthesize ATP in all living things, and these are coordinated, enzyme-mediated. They're all reliant upon enzymes, and we talked about why those were important earlier this unit. Reactions that take energy from molecules, all right? They're going to be transferring energy from one form to another, just like we spoke about earlier this unit as well. 3 stages of it. Glycolysis makes ATP and NADPH by converting glucose into pyruvate. It does not require oxygen. All right, none of the glycolysis stuff requires oxygen to be there. You can all sorts of organisms just live off of this, and it occurs in cytosol, and that's what's called fermentation. It's the production of ATP by only using glycolysis, and it makes products like ethanol and lactic acid. Okay, the Krebs cycle is phase 2. That takes pyruvate into the mitochondrial matrix and produces more NADH and FADH2 electron carriers. All right, and those electron carriers are used in phase 3, the electron transport chain, and that occurs along the inner mitochondrial membrane. And it takes those electrons from our electron carriers, NADH and FADH2, to establish a proton gradient and power ATP synthase, the phosphorylation of ADP. Using a proton gradient, okay, using chemiosmosis, just like what we saw in our last topic. All right, here's the 4 main steps of the mitochondrial electron transport chain. All right, what I animated here for you before. NADH and FADH2 deposit electrons to proteins on the inner mitochondrial membrane, and those proteins, what they do with those electrons that are getting passed around, the energy from those electrons, to pump ions out from the matrix into the intermembrane space, and that creates a proton gradient, all right, the difference in concentrations. And then that concentration gradient, that proton gradient, is used by ATP synthase, right? The ATP synthase is the way for those hydrogen ions to get back into the matrix to phosphorylate ADP and an inorganic phosphate, NP, through chemiosmosis. All right, so once again, ATP synthase is going to be using what's called that proton motive force, chemiosmosis, to take ADP and take a P and boop, piece them together using the flow of those protons. And it's able to do that in large amounts, and that's called oxidative phosphorylation. We can make 30 plus ATPs with that, okay, with the energy from one glucose molecule. All right, and then those electrons that are passed from one place to another, they are accepted by oxygen, which binds with hydrogen ions, protons in the matrix to produce water as a byproduct. All right, so that's why you need to breathe. That is it for this video. Let me know if you have any questions.